Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled In Silico, From Knowledge Curation to Clinical Trial Simulation. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Novadiscovery, who developed the content for this presentation. Nova has built the world's first collaborative clinical trial simulation platform, Jinkō, for pharma companies to bring innovative medicines to patients faster and with greater confidence. And now I'd like to introduce our speakers for today's event. Frédéric Cogny graduated from École Polytechnique in Paris with an applied mathematics master in probabilities and finance. He then worked for 12 years in financial market modeling and quantitative trading across London, Toronto, and Tokyo. So his early focus at Nova, though, was on industrializing the science of biosimulation with systemic model hypothesis testing through the prism of scenarios and large-scale simulations. Now in charge of the Jinkō roadmap, he ensures that the platform development delivers the best product experience and most value to Nova customers and users. And also joining us today is Dr. Ingeborg Friehs, who is a physician scientist at Boston Children's Hospital with a faculty appointment at Harvard Medical School. Her main focus is on translational and clinical research investigating the underlying cause of complex congenital heart defects and to develop novel therapeutic approaches. She has published extensively on these topics, which has led to collaborations with US and international academic partners. She teaches locally at Harvard University with emphasis on clinical research topics. But without further ado, let's go ahead and hand the presentation to our first speaker for today. Fred Cogny, you may begin when you're ready.
Thanks, Ryan. Hi, everyone. It's a pleasure to be here. So today we'll be talking about Jinkō, but especially how you can use it in the context of designing clinical trials. We'll have a focus on the knowledge module, which is a part of Jinkō, and you'll hear from Dr. Friehs how she's been using it in real life. So to give you an idea and an overview of Jinkō. Jinkō is an in silico simulation platform. And by in silico, we really think of in silico as a third pillar that comes and supports both in vivo and in vitro clinical trials. It has many advantages for you to run clinical trials in silico ahead of real life trials. And those advantages span things like reducing sample size, increasing trial efficacy, avoiding errors, costly backdated errors by predicting them ahead. You can address also ethical concerns. You can avoid enrolling too many patients, and for instance, by running synthetic control arms, many advantages to that. At Nova, we run in silico using the Jinkō platform. Many advantages include, for instance, you have an unlimited number of virtual patients because you're only limited by computing capacities. You have real control because each patient is its own control. You can run trials for as long as you want, so you're not limited by real life durations of 3 to 4 years, especially for chronic diseases, for instance. And you can run as many experiments as you want because you can have unlimited number of arms. So those are the few advantages of running things in silico. The way we've conceived Jinkō is first and foremost a great product experience across the entire platform and a seamless experience wherever you are. By wherever you are, we mean 3 main modules. The knowledge module that allows you to extract information, classify, curate it, classify it. Then, based off that knowledge, you will be running and designing clinical trial simulation, trying to get further knowledge by running your hypothesis in a sandbox in silico, and then analyzing those results through the prisms of analytics, doing things like comparison of result arm, applying eligibility criteria, grouping patients in different groups, comparing survival analysis of different arms. You can do that all online within the platform. So Today's flow will run in 4 main steps. First, I'll show you how you use the knowledge module to extract knowledge and classify information. Then Dr. Friehs will explain how she's been using those capabilities in real life. Then we'll move on to designing a trial within the platform, running the simulation and analyzing the results. And then we'll see how those results can be then integrated back into the knowledge that we collated in the first place to have this virtuous cycle of knowledge, hypothesis, testing, creating new knowledge, and incorporating these back into the flow. So without further ado, let's start by trying Jinkō. And indeed, I'll show you the first module, which is the knowledge module that we have within Jinkō. So here what you see in front of you is a collaborative multi-user document in which we have collated knowledge. It gathers pieces of knowledge, and those little icons here are links to extracts that we took and we gathered from the literature. The other way around, for instance, is I can extract information directly from the paper, collate it, create those new extracts. I can go and I can classify them, give them titles, and essentially giving them a strength of information. For instance, here I would have excellent data. So and here would be PK model structure, for instance. Then I can go back in my document and gather this information and reuse it by creating new knowledge directly. And within the document, you can see how each of those hypotheses that or this knowledge that you've extracted from the literature is easy to just drag and drop and just insert, and it will be part of your document that you collaborate with. So in general, you can just import any source, any reference that you have with the actual source by just browsing and inserting any document, for instance, here Nature Primer. It will extract the bibliographic references for you, create a reference, validate. And once you have this new paper online, it's collaborative with your co-workers, and you can again extract any information the same way you would be doing a highlight, creating an extract, and you can use that extract again within your document. So this is just how, to give you a brief highlight, how we use Jinkō knowledge, import sources, collaborate, extract, and classify information. So I'll hand it over now to Dr. Friehs, who will explain how she's actually been using that knowledge module in the real world.
Thank you very much, Fred. I am happy to share the next 10 or so minutes with you and to give you a little bit of an overview what I have been doing with Jinkō, or what does Jinkō mean to me in general. I have no disclosures to make pertaining to this presentation. And this is just a brief overview how my talk is going to be presented. So a brief introduction, the uses of Jinkōs, and then go a little bit into detail about the advantages and disadvantages of this platform. So the first question that I asked myself when I started preparing for this webinar was, so kind of what can Jinkō do for me? And I think the primary question that I asked in order to find something like Jinkō was, how can I better use my time? And as you're just like me, time is our most precious asset. So at least I am always chasing after making my life a lot easier, because the day only has 24 hours. So in order to guide you a little bit further through my use of Jinkō, what I would like to do is to introduce myself to you. Introduce myself to you as the human being that I am and what I spend my time with. So the real me is a principal investigator of translational and clinical research projects, an applicant. So research requires funding, an applicant for funding, for extramural funding. I'm an academic teacher, so this is part of my responsibility to educate the next generation of physicians and scientists. I'm a mentor of these people. I'm a supervisor of the people that I'm responsible for teaching, as much as I'm also a supervisor for everybody else who is who I am responsible for. And I'm a manager of all of these people. So I give you the idea of what the team looks like. So the team are all the people that I interact with on a regular basis. So these are postdocs, fellows, these are students, can be medical students, can be master's students, administrators, somebody takes care of my grants, somebody takes care of the finances, regulatory entities. Just to give you one example here, the IRB, so the Institutional Review Board, or the Ethics Committee, as it is known in other parts of the world. Funding agencies, in this case, it's NIH. So all of these interactions between me in all of these roles and the team are very frequently on a one-to-one, either in person, on Zoom, telephone calls, lots and lots of email exchanges, meetings with meeting notes, and everything is filed away or half read, but it's all in separate aspects and in separate folders that all of this information is stored. So a brainstorming session for a project. This is the kind of the top of the iceberg. And I just broke it down into the two main aspects where you spend a lot of time with. So one is getting a research project up and running, and the other one is the completion of it, like the publication. And the team and the interaction with the team is in the center of this, and it's a back and forth. So it's not a one-way road, but it's a back and forth. So what do we do with regards to all of this knowledge that we are compiling here? The knowledge that we are compiling here, that we are collecting by reading, by searching PubMed, then reviewing, editing, and then ultimately creating a publication. So we all know reference managers, all different kinds, and I don't have to go into detail, and I'm not going to mention any of those. So imagine reference managers. You just basically passively store your PDF and your information, and then you have to go back and reread. Yes, you highlighted the necessary information, but yet again, it's always a back and forth, and it's always a passive kind of interaction. So now this is where Jinkō comes in. And I just highlighted for you here what Jinkō can do for you. So a pure PDF is no longer just a passive platform, but you can import and export. You can highlight, you can edit, you can discuss with your team, because it's an interchange. You can reference, and even more so, like in my role as an academic teacher, as a mentor, as a supervisor, what I can also do is kind of keep track of knowledge curation and also knowledge compilation, and being up to date with the most recent scientific advances, because it is a shared platform that everybody who is introduced into this team can participate in, and I can also keep track of this aspect. What can you do on a study design aspect? So with regards to study design, there is a little bit of a different kind of interaction that is necessary. On top of the scientific and the knowledge aspect, it also can be, Jinkō can also be used as a communication platform, because the interaction with my administrators for document exchange is a different one than with my team members who are responsible for curating scientific knowledge. I can open this platform to everybody who is part of the team, and there are no restrictions. So I do not have to worry about somebody using a different software platform, different software, different computer platform, but it's all in one, and it's an open access for all team members, but secure with regards to information contained, and we can exchange documents, we can exchange information, and we can also track and store all of these documents and all of this information. So basically what it comes down to, what for me the usage of Jinkō has facilitated is the managing of documents, the managing of data, the managing of people, and it's in a very standardized format without having to go back and forth between different platforms, and it makes the workflow much more streamlined. So obviously everything has advantages and disadvantages. I've already provided you with a lot of the advantages from my point of view. So the integration of the word processing, a reference manager, and a file storage platform all in one. It's user-friendly, you can extract PDFs, it's immediate access for team members, and there's no storage limitations, and it's an open platform for import and export. Limitations, there's a learning curve, but this is true for every software program that you use for the first time. Also a disadvantage, it's only offered as an online version, so exchange can only be done online. It is not open source. It has no browser extensions. It has no mobile view or app. And the PDF extracts can be saved with the PDF, then cannot be saved with the PDF, then be exported. So in general, in my opinion, my life has made easier with a platform like Jinkō, and I'm looking into the future, what else Jinkō can offer to me, because so far this is what I've been using it until now. Thank you very much for your attention, and I would like to hand it back to Fred.
Thank you very much, Dr. Friehs, for this great feedback. You'll be glad to know that actually on the stable internet connection issues, we made recent progress, and if you get disconnected while you're on a train, for instance, on transport and working on your document, all your changes will be saved and you'll be able to keep editing. Obviously, you won't be able to launch trials in the cloud, because to launch things in the cloud, you need a connection, but stability of the documents have improved. And thanks for the other feedback. We definitely take these into account into the roadmap design process. So this is great. So following up on that, once you've extracted knowledge in a collaborative way, the next thing is, what do you do with that knowledge? Dr. Friehs has mentioned a few things like study design and the way you can brainstorm with others. Having an in silico sandbox allows you to do that. So the next step is we'll show how we can design a trial based on that knowledge. Then we'll analyze the results and again include those results back into the knowledge. So just for a little bit of scientific background and context, I'm going to run a trial on a non-small cell lung cancer model. And so this is an in silico mechanistic model based off knowledge extracted from the literature that captures tumor heterogeneity intra-patient, but also captures the inter-patient variability with a virtual population. So we'll show you during the demo how we can test different hypotheses, not just on one patient, but across a variety of patients by running a full trial on a virtual populations. So going back to the document, which doubles, just as Dr. Friehs explained, as a reference manager, we can see all the references here that were being used in establishing the pieces of knowledge useful. We have here a presentation and an overview of the NSCLC model, for instance, that I would write as a summary. You have, by the way, some handouts that come in this webinar, and one of them is a flyer of the NSCLC model if you want to get an overview of the model capabilities. So Some of the questions that this model can address are things like the impact of various mutations on the trial results and on the response of a treatment, which was gefitinib here, which is modeled with a treatment. Another one we can test for, and those are two questions we are going to be addressing today, is the ability to test different doses. So I've listed the questions here as if it could be done in a separate document. Here we'll be doing in the same document, and we'll be doing the various steps of creating a virtual population, creating protocols, and then analyzing trial results. So let's start by a first step of the simulation, and let's create a new virtual population that is based on the NSCLC model. And we start by designing the properties we want that population to follow. We start by introspecting the model, and we see all the various inputs that we have within a model. We have things like age and so on. And so we can generate a patient, and here we'll generate a population of 100 patients for the purpose of the trial. We give it a name. So here, full virtual population, based on the properties we want our population to follow. We can see the distributions of each of the patient attributes. And once we have a population, the next step is to naturally create a clinical trial. A trial is the combination of several elements. Your computational model that contains both the disease and the treatment, a virtual population, here are 100 patients that we just designed, and on top of this, we're going to be running a protocol, and we'll be doing some analysis on the outputs of the trial to compute the quantities of interest that we want to look at in the context of our trial. So let's start by creating a first new trial, just to test, for instance, the first question, which would be the dosage. So let's say a protocol to answer the question 1, impact of dosage. We'll be selecting descriptors that are of interest for us, such as the dose, for instance, and here in human, as well as whether we have a placebo flag or not. And we can here create as many arms as we want. Let's start by creating one where there is no treatment, which is the natural disease course, for instance, with no treatment. And then another arm where I'll be having a dose of, for instance, the regular dose, which is a default dose of 250. We compare that arm with the disease arm. We can yet create another arm, but this time we have, let's say, a higher dose of 450. So again, we can create as many arms as we want. We're not limited, and we can test different doses. Very simple this way. In the platform, everything is linked. So when I create a protocol which is linked to a computational model, I see the link appearing here. When it was initiated from a trial, I can see the link here again. So the transparency and ability to navigate easily the platform is crucial. And I can go back to my trial, see that the question 1 protocol is picked up, and so on. Another thing we want to do is compute quantities of interest. So I preset some quantities of interest here in the mesh design. I'll show you what those are. They are give you the ability within the platform to directly compute summaries over the full output. So for instance, if I want to look at the maximum value of the tumor radius across the entire simulation, it's an easy thing to do. Same thing on the tumor radius, this time the maximum value, but I will compare it with the control arm. So it gives me the quick ability to compute quantities of interest, clinical values of interest, and associate them with my trial in terms of post-processing. So now I have my output that will compute, my protocol, my 100 patients, and my model. I'm ready to launch a trial. So I have my 300 patients to solve. So once you're rerunning the trial, which will be relatively quick, only 2 minutes, as you can see here, one thing that I started talking about is the transparency, auditability, and navigation. My trial here was linked to a virtual population. I could see my virtual population was linked to population design. I could see that my population design was based off the computational model, and so on and so forth. I can go and analyze and introspect all the links. Same thing, I can see that my computational model is referenced into a document, which is a document I was showing you earlier. And I can go back to the document where my model is referenced. So I have full transparency on the chain of information that allowed me to establish a given results. One thing we do as well in the documents, beyond drag and dropping pieces of knowledge that we have, is we can include any of the project items that are being present within our project. So we go beyond the information extraction and classification. We can document any of the items present in our project, including, for instance, we can document subparts of my model. So here as an example, I chose 4 parameters of my model. I could select more than that I could render. Those are all the model parameters. And I can go and introspect their definitions. I can see what it depends on, what references they are on. And I can see even those references, again, are pointing to pieces of literature that I can open in context. So I can see what the model is made of. I can see what justifies its construction, and I can go back to the elements. Traceability is crucial within Jinkō. So my simulation should be complete by now, and I have the ability to maybe answer the first question and look at trial results by looking at the simulation. So trial is indeed complete, 100% completion, and I can create a visualization of my trial. And I can compare the efficacy, for instance, at the population level across different arms, disease, and treatment, and I can compare the efficacy at the population level. So I can see that the time to progression is null. That means all patients have an increase in their tumor radius right from the start because they're not treated, and I can compare my 2 arms with and without treatment. I can also compare the time series of those 2 outputs, and I can compare the tumor radius, for instance, by selecting just the 2 arms that I want to compare, for instance, 250 versus 450 arms, and I can analyze just those results. Another way, especially in oncology, to compare results, I've got the 2 time series. I can see there's a mild higher efficacy, essentially for treated 450, because the tumor radius is slightly lower across the entire population. Another way to look at it is, again, to do the time to survival for the time to clinical progression. I can run the survival analysis on the 2 groups that are selected, but I see just a marginal difference between the 250 and 450. So the marginal increment, the marginal value of increasing the dose, for instance, might not be that high, and 250 would be enough. That's, for instance, to answer the first question. So to go back to the document, we can insert those results in C2 in order to document the first findings that we had. And the same way we were documenting extracts, I can start putting here for my colleagues a link to the virtual population, the protocols, and the items that I've been using to document my first findings. So let's start by documenting the virtual population. I can then start documenting the protocol for question 1, and I can insert a link to trial results and the visualizations so like people can go and have a look at it. But I can also, one thing which is great is you can insert in your documents essentially results that you've been incorporating. You can just download those results and insert them back into your documents and the results. So now let's explore another question, for instance, a different way to explore the powers of protocol beyond what you could do in real life. So let me just include those results. Sorry for that. I'm just gonna, there you go. I've got a survival and I can document here the results in CG. The second question we want to ask ourselves is, how do specific mutations impact the response to the treatment? So one thing we can do in real life is test different doses, but we cannot go and change people's mutations. But because you're in silico, it's an idealized situation, you can test those hypotheses within a given trial. So let's run another trial here, where we'll be testing the same hypothesis. And I start again from the same population, but this time initiating a new trial on the exact same population. And this time I'll be setting a protocol for question 2 that I've set up. I'll show you what it looks like in a second. And again, computing the same quantities of interest. The protocol design here in this instance is one where I can play with any of the patient characteristics, and I have one which is still the disease with no mutation. One arm where I have the treatment but no mutation, and then I activate selectively on each arm the different mutations. So I can play and ask myself questions ahead about whether I should include or exclude patients with certain mutations, and use these in my study design as eligibility criteria. Again, I have transparency, and I can go back to my trial, open a trial, and then ready to launch it. So in just a space of a few minutes, this time I have 600 solving because each patient is cloned across 600 arms, 6 arms, sorry, so 600 patients. And I can run, again, similar situations. A lot of the patients were exactly the same as in a previous simulation, so it's super fast to solve this time because it's clashed. And because we're in a cloud and the grid is already warmed up, it's much faster to run the exact same simulations a second time, because we have many computers at our disposal. That's one of the advantages of running things in the cloud. Post-processing my result, I'm now able to create visualizations and directly compare the results of the second question. And here this time, I can compare. Let's just focus, for instance, on the radius and compare the different arms. And then just select all arms but the disease arm to compare the output. And I can see the distribution of the time to progression across different arms. And I can see definitely when they have no mutation that it's the largest time to progression on average. And I can also see that the one with the Curation mutation is the lowest. And I've got a higher average, for instance, on BRAF. So it looks like Curation, for instance, has a negative impact on the treatment efficacy. Another way to confirm it is, again, to look at the time series and compare different outputs. So here, let's just focus on tumor radius and data results. And I can compare all my arms, and I can just easily activate them and deactivate them. So we are focused, for instance, on KRAS as one of the mutations versus the no mutation. You can see a massive impact, negative impact. Essentially, the tumor radius is much higher for patients with KRAS. And again, another way to look at it is a survival analysis, whereby we can select our time to progression, clinical progression, and compare the impact on survival curves across the different arms. Again, we can see that the no mutation is the one that has the longest survival, longest expected survival, whereas if we compare the different mutations, KRAS, for instance, stands out as one that has the least survival in terms of curves. So KRAS typically would come out as an eligibility criteria for exclusion to include in the design of the trial. So we can, again, download the results, include them into our document, and confirm to our colleagues that we should include that mutation as part of the exclusion criteria. I go back to my document and include my findings in my results. here for the second protocol as part of the elements present within my project. So this is it. There we go. And quantity of interest. I include that. Sorry about that. Quantity of interest, and I include the results as well right after that. And the findings here will be inserted in the document. So this is it. This ends the demo portion of this webinar. Again, we've been covering the 4 steps. We've been extracting knowledge from the literature using Jinkō knowledge. Dr. Friehs has explained how she uses that in the real world. Based on that knowledge and the fact that we distinguish KRAS as a potential impact, we've been able to design a trial, first on the dose, and another one distinguishing different mutations, whether to include them or not as inclusion and exclusion criteria in a trial. We've been able to run a trial in Silico extremely fast, analyze those results, compare different groups, compare different arms, and based on the outcomes of this trial, analyzing those results. And we know that including those results back into the knowledge, we're able to collaboratively share with our collaborators and show our findings and put digital evidence as to what we find. And here I can go again and I can, for instance, comment and share with any of my colleagues the findings that we have, and in a collaborative way. This is the comment portion. I can just tag any of my colleagues, for instance, present in a project, to have an opinion on this. You can send and receive a notification and you can act on it. Here we go. In the scientific context. I think we now move on to poll questions. Ryan, if you can take the lead on that. Thank you very much again, everyone who's attending. I hope you appreciate Jinkō, the presentation. Thanks also again, Dr. Friehs, for your explanations on the knowledge.
Yes, well, thank you both very much for this presentation. As we continue, though, today, we do have a poll question for audience members. This should be appearing on everyone's screen right now, and you can participate by selecting on any of the answers you see in front of you and then clicking submit. The question that we're asking is in regards to Jinkō knowledge, among those features, which one would you like to see coming first? NLP enhanced scientific watch, ease of publication, or plagiarism checking? We'll give everyone some time to consider their answer as it best applies to the question again of among those features, which one would you like to see coming first? Looks like we have a majority of the answers in, so thank you very much for your participation. Let's take a look at where your results came in for this question. We have 50% of you selecting NLP Enhanced Scientific Watch, 29% for ease of publication, and then 21% for plagiarism checking. Thank you very much again. Let's move on to our next poll question here. Should be appearing on everyone's screen at this time. Again, please select an answer and then click submit in order to participate. The question this time is in regards to Jinkō simulations. For modelers regularly refining and editing their models, what would you consider to be most important? Your answer options are import your models from Symbiology, R, etc. into Jinkō, create, edit, and calibrate your model directly in Jinkō, or just an NA option to say that you're not a modeler. In a moment, we'll hear from our speakers some more to answer some of your submitted questions. But the question we have for you right now is, for modelers regularly refining and editing their models, what would you consider the most important? Let's take a look at your answers for this one here. 44% selecting that you're not a modeler, but then we have 28% split between importing the models and then creating and editing the model directly in Jinkō. It's a very interesting answer there. I believe we have one more right here for everyone. This time, what we'd like to know is your usage. So among the various Jinkō modules, which one do you see yourself using the most? Your answer options are knowledge, trial design and simulation, analytics, or you have an option for all of them. Please continue to send your questions or comments right now, though, through the questions window for the Q&A portion that will start shortly. Also, you can look to the handouts module within the GoToWebinar control panel, where you'll find some additional documentation related to today's presentation to download for some further reading. Again, the question we're asking you right now, though, is among the various Jinkō modules, which one do you see yourself using the most? We have most answers in now. Thank you very much for participating. Let's look at your results here. Look at this, 53% for all of them, and then 29% for trial design and simulation, 12% for knowledge, and then 6% for analytics. Wonderful. Thank you very much, and thank you, our speakers, for the presentation, this wonderful presentation. As I said, we're moving to the Q&A session. We've already received some questions from our audience, though, so we'll get ourselves started with those. The very first question that I have for you would like to know, what's the difference between AI and NOVA's methodology?
Thank you, Ryan. So NOVA's methodology is primarily based off knowledge. As you've noticed, we extract knowledge from the literature. But then when it comes to calibrating your model, we have a hybrid approach between AI and knowledge-based mechanistic approach. You need data to calibrate your model, and you need calibration algorithms. Here we use, for instance, Bayesian calibration algorithms that leverage machine learning type of techniques to help us essentially formalize both constraints we have extracted from the literature, as well as objectives in the form of data that we can fit into our calibration algorithm. So we use a hybrid approach when it comes to calibrating our model. One of the questions earlier as well that we saw, that you heard, was, do you prefer NLP enhanced, or do you prefer to have personal space and things like this? So in the NLP space, when it comes to suggesting potential relevant articles, we'll be using AI there too, to identify the themes that you're working on, the keywords you're working on within your project, and suggest relevant articles of interest. That's one thing. And the second thing is, once you import a reference or a source, to potentially highlight extracts that could be relevant for your research as well. The goal is not to supplant humans, but to really assist as in a cockpit version, to make you more efficient, to make you save time, just like Dr. Friehs was saying, and make your life easier, but not replace you. They won't do the analysis for you. They will help you get there faster. That's how we want to use AI in our process, essentially. So it's not as opposed to, but it's really how do we integrate AI within a process. Thank you, great question.
Yes, thank you so much for that answer. That was excellent. We've got another question here wondering if this is open access, and is there any trial license available for use?
So thanks for the question. So it's, we have open access for all academics, unlimited. If you are from the industry, we have a free trial period. You just contact us. We create an account. We can do onboarding. We have also a customer success team who's here to help you get onboarded within the platform. We have two things, right? You have Jinkō, which is a platform, which is one thing, it's the toolbox if you want, but you also have the content. So Nova offers three things: the toolbox, Jinkō, content open for subscription in a form of models. So in this demo, I was showcasing the use case of NSCLC as one of the models for subscription, available for subscription, sorry. And you also have managed services. So if you're in the industry and want to get access to Jinkō, but want to work on a specific program, our team of scientists and biomodelers can help you answer those questions, tailor a model for you. That's part of a third offer. So it's open access if you're academics. If you're in the industry, we can create trial account for you as well. So it depends in which case you are, but both are available.
Good to know. Thank you so much. The next question that I have here wonders if the in silico analysis is leveraging metadata from the current literature and then assumes that published trial population reflects the general population.
That's a great question. So You've seen in the demo that we have the ability to design any virtual population of interest, and we can take the ones that have come with a model that are set by the modelers for you within the platform. So when you subscribe to a model, you don't get just a model, you also get the analysis that comes with it. You get a default virtual population. You also get default protocols, the documentation that comes with the model. So it's a whole package. The population, which is within that package, has indeed been fit to the general population that is inferred and from existing cohorts, essentially, and that are relevant for the disease of interest. So they are indeed tailored to represent the general population, the target population for the indication.
Right, very good, thank you. The next question just simply would like to know, do you need a lot of data?
The more the better. We always do need more data, but not that much. Again, linked to the previous point about AI, we have a hybrid approach where the structure, the core of the model, the way it's built, is really based off knowledge. So we don't try to find data to infer what a structure would be. The structure comes from the knowledge. And the data is used in the calibration phase and the validation phase in two separate steps. The way we also calibrate uses two types of data: structured data, and that's usually what people think about when you mention the term data. They think of tables, databases, but there's also unstructured data. If you think of publications, all the text on the knowledge that's captured in the papers is a form of data, unstructured data that is, but we extract it and we formalize it in the forms of constraints the model has to follow. So at the end of the day, we don't need necessarily as much structured data, and clearly way less structured data than a typical AI model would. But we do need data indeed to calibrate our model, but in an order of magnitude way inferior.
All right, very good, thank you. So next, I have someone wondering, how do you validate your models?
So it's the several-step approach. Our models are based off knowledge, and we start by creating that knowledge. You can't be an expert in every therapeutic area. And so when we work with clients and when we design the models that are open for subscription, we work with external experts specialized in the domain and the therapeutic area. They will come and validate or invalidate our findings, or tell us, oh, you've missed that paper and you've missed that publication, or that hypothesis there is strength of evidence judged too high, for instance. So that's a first form of validation, is the knowledge that we have sufficient, up to date, and covers the whole spectrum. Then when it comes to the formalization of the model, we have both visual and qualitative checks that our model behaves appropriately. And then the most stringent is really quantitative and data-driven, and this is where having data becomes important. And the validation of the model is based on a separate set of data that was used in the calibration process. So first is qualitative on the knowledge, then qualitative on the model, then quantitative on the model itself.
Perfect, thank you. Next, someone's curious if they can reuse your models for their own applications.
Absolutely. So our models are open for subscription. They can be used with a different treatment. So here the example was the standard of care was gefitinib, and the models can be either available with different treatments, or if not, we can enhance the models, create a new version of the model to plug in different treatments that can be plugged into those disease models to test, again, different doses, different treatment combinations, your treatment with the existing standard of care, and so on and so forth. So that is totally doable.
Very good. The next question is curious if regulators, if they're in favor of the in silico approach.
They definitely are. They are in favor, they are encouraging, they are looking at it themselves. And it's been voted, I think, in the Senate to encourage the use of in silico in submission dossiers at the FDA. So there's an encouragement, and there's a whole expertise within the regulators themselves to look at in silico the ability to replicate the findings. They asked, for instance, for sending them the models to sending them the findings, and so they can replicate the trials themselves. In this vein, Jinkō offers a great advantage. Not only will they be able to use the platform as regulators to come in and launch simulations themselves, replicate results themselves. On top of this, they have the full transparency that the white box approach that Jinkō brings offers. So they'll be able to see existing results, trace back, oh, this trial was designed this way, I can see the protocol, oh, this protocol was based on this computational model, and then this model was based off this knowledge on those papers. So the ability to trace within a platform will be a great advantage, we believe, for the regulators.
Very good. A big question here wonders if Jinkō answers FDA's Project Optimus requirements.
So you can. So the Project Optimus, for a little bit more background, is how do you do first in human? How do you essentially handle the translational part? And as you've seen within Jinkō, you can test different hypotheses, different dose, you can do dose escalation. One thing which is interesting is currently in real life, you do 3 + 3 + 3, so number of patients you recruit as you go and you do dose escalation. In Jinkō, you don't have that ethical concern, obviously. So the moment your model is validated, you can test dose escalation all in parallel, and you're not limited to 3 patients. So the Project Optimus is how do you enhance the current methodology, and Jinkō is definitely one of the solutions that can be brought to the table. Obviously, you need your model to capture the toxicity, but once you have that, the ability to run trials in parallel and to go well beyond the limitations of 3 real-life patients is something that Jinkō addresses.
Wonderful. Thank you so much for that answer and for all the answers. However, we have reached the end of our time here today. Now, if we couldn't attend to your questions, though, the team at Novadiscovery will follow up with you after the webinar. I want to thank everyone for participating in today's webinar, and you'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I've also just sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues once they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you, Ryan. Thanks for everyone for attending. Thank you, Dr. Friehs.
Thank you very much.